Hello everyone, my name is Ritu Sharma and I am a research fellow at Pine Biotech. Today's presentation is based on my project, which is on the topic surveillance of SARS-CoV2 variants of concern from wastewater samples. The objective of my the objective of my project is to learn and analyze variants of concern in sample and perform MSA and phylogenetic analysis using NGS data employing T-Bioinfo server. These following are the list of contents which are involved in the today's presentation. So based on the learning which I have done during the course is Introduction to Bioinformatics. Bioinformatics is the application of computer technology and statistics to the management and analysis of biological information. In this, we gather, store, and analyze and integrate biological information. Many of the recent groundbreaking discoveries in biology, chemistry, environmental science, and other research areas are rooted in bioinformatics. Big data is a collection of data and is in huge volume, which is used for data-driven discoveries. Data-driven discoveries mainly include 6 steps, that is identifying question, second, review findings, third, designing model, fourth, gathering data, fifth, analyzing data, and sixth is implementation of the result. Bioinformatics has been proved to be a boom in many industries, in many fields like healthcare industries and agricultural industries. So in healthcare, it is helpful in making precision medicine, drug discovery, preventive medicines, and gene therapy. Translational bioinformatics is also an emerging field which brings biological research into clinical significance in both patient care and drug discovery. Next learning is about the R coding, which was learned for pairwise sequence alignment and multiple sequence alignment. So pairwise sequence alignment means aligning two sequences and is also the basis of database similarity searching and multiple sequence alignment. There are two methods of PSA, that is global alignment and local alignment. On the other hand, multiple sequence alignment is the method used to compare multiple long and short sequences to find matching portions, introduce gaps, and find consensus sequence. Here in the two pictures, which we can see on the slide, tells us about the different codes for PSA and MSA respectively. First, we need to load the libraries, then we need to load the data string, and then we need to give a command for pairwise alignment or multiple sequence alignment accordingly. And then the last command is for the printing the alignment. Genome sequencing. There are mainly 3 genome sequencing which I have learned. So the first one is targeted NGS sequencing. It is a rapid and cost-effective way to detect known and novel variants in selected sets of genes or genomic regions. Whole genome sequencing provides a comprehensive picture of information in the genome by reading both coding and non-coding sequences. In short, it reads the whole genome from start to the end, involving the non-coding regions and the coding regions. WGS generates more detailed look at the information of our DNA. Whole exome sequencing, on the other hand, targets only the protein coding regions of the genome, providing less coverage, and only gives us the information about the coding regions of the genome. Variant calling refers to the task of identifying possible variations in genome or transcriptome sequence with respect to chosen reference sequence. So it tells us about the different variations in the genome with respect to the reference sequence, which helps us to identify the mutation in the reference genome according to the reference genome in the sample. So there are two types of variant callings, that is germline variant calling and somatic variant calling. So germline variant callings are, these are the variations which are present by birth and does not get transferred from one generation to another, and are usually taken as the reference sequence of the standard species for this species of interest. Somatic variant calling, on the other hand, are the variations which are developed with the course of time and does not get transferred from one generation to another, and they are usually taken as control somatic cell samples. The whole genome is read in two methods, that is by paired end reads and by single end reads. So in paired end reads, the fragment is read from both the ends towards the center of the fragment. In single end, the sequencing fragment is read from one end to the other towards the another end. Here is phylogenetic tree, which we can see it's a dendrogram. There are many different tree representations in phylogenetic analysis, but here we have taken the dendrogram. So phylogenetic tree tells us about the closely evolutionary relationship among the species and their origin. So here in the diagram, A, B, C, D, E are referred to as taxon, and the branch which is emerging from a common origin, which is the node, gives two clade, that is C and D and A and B. So C and D is in one clade, which tells us that they are very closely related to each other. If they are in close relationship with each other. Mutability in simple terms describe the observed rate of given position to the change or mutate. Differential mutation is an evolutionary algorithm that processes a population of individuals represented by dimensional vectors of real numbers. Copy number variation is a type of variation in which a section of genome is repeated and the number of repeats in the genome varies between individuals in the human population. Here are the different pipeline graphs which I have learned during my course to identify different things like variant calling, mutability analysis, phylogenetic tree analysis, and copy number variation. So different pipelines are used for analyzing the result for different things. The pipeline I used here for my result and interpretation with the data set which I got from NCBI for surveillance of SARS-CoV2 variant of concern from wastewater sample, which is my project title. I got this sample for wastewater samples from US and UK, which were taken as control, and I have taken the reference sequence as the severe respiratory coronavirus isolate from Wuhan. The pipeline graph, which is shown in this slide, was performed, that is for phylogenetic tree analysis, and the result I got is this tree. So the result interpreted is the sample ERR5019830 and ERR5019834 are in same clade in the phylogenetic tree and have closer evolutionary relationship with reference genome. Since common node for both clades, sample ERR5019835 and ERR5019840 are in the same clade and also share closer distant evolutionary relationship with outgrowth sample. In the phylogenetic tree, the sample ERR5019836 and ERR5019838 are in different clade and which does not have closer evolutionary relationship with the reference genome or outgrowth sample, as they are in different clades in the last. This research paper is based on my topic, that is surveillance of SARS-CoV variants from wastewater, which I considered to read in my project. So it is on monitoring SARS-CoV2 population in wastewater by amplicon sequencing and using the novel program SAM Refiner. In this research, SAM Refiner program was developed and used to find out the variants of concern in wastewater sample. So as a result, two variants of concern were reported, that is alpha and gamma. Thanks. This was all for my today's presentation. Thank you.